So in this next video, we will be concerned about the building blocks for DNA and RNA. And keep in mind that DNA and RNA are polymers that are usually very big. And the monomers, the constituent parts of DNA and RNA, are known to be called nucleotides. And that's what we see here. So nucleotide is made of basically three different molecules linked together via covalent bonding. So the first part of this nucleotide, so the first part of any nucleotide is something that we call a nitrogenous base. So the reason why it's called nitrogenous, I think you've guessed it here, when you look at all those nitrogen atoms that are part of this molecule here, this one here, well, that's why it's called nitrogenous base. And this base here, this part here has a name, and we'll see there's few different ones, and this one is known to be adenine. And the abbreviation for adenine is A. So this adenine is linked through an oxidic bond. Remember what an oxidic bond is. It's connected to this sugar here, and this is the sugar in blue right here. And the sugar in this case happens to be ribose. And we'll see that there's another kind of ribose, another kind of sugar rather, that's called deoxyribose. And then every single one of these sugars, when we're talking about the bigger molecule, are going to be linked through a phosphate group. And if you remember well, when I was talking about lipids, I was saying that lipids, some of the phospholipids, have a negative charge. Well, that's because of the phosphorus right here. The phosphate group, as we see here, has been known to be charged negatively. Now, if we concentrate on the nitrogenous bases, we can see that there's diversity in those bases, and those bases are part of two categories. So on top right here, we have the pyrimidines, right here, pyrimidines, and you see that the pyrimidines only have one cycle, and there's two different pyrimidines. One is known to be cytosine, and the abbreviation for cytosine is C. We also have thymine with the abbreviation that is D. Now, look at those two. They kind of look very similar. They contain a lot of nitrogen atoms in every single one of those molecules. But when you look at the tiny differences between cytosine and thymine, you can tell that in thymine we have this chemical group here that has been added in this case. Now, when we look at the other ones, the purines, and the purines are the more complex nitrogenous bases, we have guanine with the abbreviation that is G, and also adenine, as we've seen before, with the abbreviation A right there. And we can see that there's two cycles for each of those molecules. We have cycle 1, cycle 2 right here, and then cycle 1 and cycle 2 right there, right? And we see that when we look closely at those two molecules, yes, they do look kind of like similar, but there are very important differences as well, and we see some of those differences right here. I'm not going to get into those differences right now, but this makes a big difference when we look at the polymer level, and when we look at DNA and or RNA. So, I know that students sometimes struggle to remember which one is a purine, which one is pyrimidine. Well, here's my take on it, and it works for me. I don't know if it's going to work for you, but this is how I remember. For one thing is that I know that purine versus pyrimidine, well, purine, they have the smallest amount of letters, the simplest word, so therefore if it's so simple, well, never anything can ever be so simple. So because of that, we have the more complex cycles. and pyrimidines, of course, are the other ones, the simplest of the two. Now, when we look at cytosine, thymine, guanine, adenine, how do I remember which one's a purine, which one's a pyrimidine? Well, the ones that have a Y in them, cytosine, thymine, of course, are part of pyrimidines because pyrimidines is the only one that has a Y in it. So anyway, that's my trick to remember which one is a purine, which one is a pyrimidine. So now if we look at this picture, we can see that we have a molecule that is probably not unknown to you. Is the famous double helix that we see here. So it's double helix because we can tell right here this molecule is twisted in space, and we say it's double basically because it has two strands. As you can tell here, there's a strand starting right here, and there's a strand starting right there, and those two strands are united in the middle right here. Just imagine like a spiral staircase, for example, and those would be the steps, right? So the two strands are linked in the middle by the nitrogenous bases. And you see in this picture right here, we see that these have been color coded for you so that it's easy to understand. So when we look at the red ones that we have here, we know that the red ones are adenine. We know that the thymines here are more or less like in yellow. In blue we have guanine, and we have guanine in blue, and of course we have cytosine here in green. So we have the nitrogenous bases here in the middle, and remember that those two nitrogenous bases, there's many of them, and those nitrogenous bases, via their interaction with one another, they will link those two strands. Now, how do they link the two strands? Like, how is that possible? Well, they will form something that is called a base pair. So what it means is that bases will link with one another, a base on one strand of DNA. So we have one strand right here. So let's say that we have strand number 1 right here, and we have strand number 2 right here. Well, the bases in strand number 1 that we have here, which are thymine and guanine, they are linked to another base on the other side, and the bases on the other side are adenine and cytosine that we see here. The interesting thing here is that we talked about pyrimidines and purines on the previous video part, and we know now, and you should know by now, that thymine will always pair. Thymine here will always pair with adenine, and guanine that we find here will always pair with cytosine. So basically, we always have those two combinations. We have thymine linking with adenine, and we also have guanine linking with cytosine. The other thing is that, as you can tell here, if you pay attention, is that we know that there are always a pyrimidine interacting with a purine, right? If you look here, we have a pyrimidine linking, so the pyrimidine is right here, linking to the purine that is adenine right here. And it's always T and A, G and C. Now, the other thing that I want, before I get into the magic of this molecule, is that I want to show here that we have our sugar right here. So let me just draw a blue circle around the sugar. So we have one sugar here, we have one sugar there. And the interesting thing here is this. What we have is something that we call sugar phosphate backbone, right? The backbone is basically the strand that we have right here. So this strand here, strand number 1 or number 2, this strand is linked, is maintained, is rendered possible via the interaction of a sugar and a phosphate, a sugar and a phosphate, and so on and so forth, right? So we will have sugar, phosphate, sugar, phosphate, sugar, phosphate. But the interaction between the two strands, so this is the link to form the one strand, but the interaction between the two strands is mediated right here in the middle, and where we have the interaction with thymine and adenine. Now, the interesting thing and the most important part of this is that those two bases are linked together, and they're not linked together via any covalent bond. They are interacting with one another via hydrogen bonds, right? So there's basically no real connection. There is an interaction, but there's no real connection there. No physical connection, I should say. It's an interaction that is mediated, rendered possible via the interaction of the nitrogenous base and the hydrogen on each side. So you see here we have the oxygen, and we know the oxygen can attract the hydrogen on the other side. Oxygen here, hydrogen there, oxygen here, hydrogen there. And the other thing that you know, and you will know by now because I'm going to tell you, is that it is possible for nitrogen to form some hydrogen bonds as well, and we have those hydrogen bonds right here. So, what's important for this here? A few things: 2 strands for DNA, bases interacting in the middle, maintaining these 2 strands connected with one another, but the connection is not physical. There is no, and what I mean by that is that there is no exchange of electrons or in any way, so it's not covalent bonding, it is hydrogen bonding. The other thing that's super important is that those bases interact with one another. Adenine always interacts with thymine. Guanine always interacts with cytosine in the middle. In the previous picture, I showed you the basic structure of DNA. So basically, we had the nucleotides, and we know that nucleotides are made of a base. They're made of a phosphate and a sugar. And we know that when we look at the strands of DNA, so when we look at the strands like this, so was strand number 1 and then strand number 2, we know that those strands are united together via an interaction of nitrogenous bases in the middle here. And we now know that A is always interacting with T, G is always interacting with C, right? Now, when we look at the basic structure of the nucleotides, where we have a base, that base is connected to a sugar, that sugar is connected to a phosphate, well, that sugar right here can be either one of these. It can be either ribose or it can be deoxyribose. Now, if you are constructing a molecule of DNA, you will have as a sugar, you will have deoxyribose. RNA will be made from ribose. So in that case, we have RNA for ribonucleic acid. So Na is nucleic acid. And then DNA for deoxynucleic acid. Deoxynucleic acid coming from deoxyribose. And the difference between the two of them is kind of subtle, really. You cannot, just as a first glance, it might be difficult to tell what the difference is between ribose and deoxyribose. It's a small difference that makes all the difference in the world. It's just that if you look at this here, the H and the OH, well, you lost the oxygen right here in the case of deoxyribose, so therefore it's called deoxy because you lost the oxygen. So believe it or not, this is a molecule of DNA, and it's presented at its the bare minimum. So what we see here basically is just the backbone of the double helix. I will make it a bit more complex and more pleasant to look at in a few seconds, but I just wanted to show that what we see here is basically is the backbone, and by backbone I mean sugar, phosphate, sugar, phosphate, sugar, phosphate. So basically what we would see here is the only covalent bonding between the sugars and the phosphates. So in the middle, we know that right in the middle of this molecule right here, right in the middle here, we are supposed to have some kind of interaction between the nitrogenous bases, right? So let's look at that right now. So let's look at what this would look like if we could see the bases. So when we look at a more realistic representation of DNA, we have something like this. We have the two sugar phosphate backbones, and I'm going to make it stop spinning for a second just so that we can see this here. What we have here, and if you follow the arrow, we have right here a twisting molecule. So if you can follow the arrow here, we have sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. The red structures here are oxygen, but the yellow ones in the middle there are phosphates, right? So we have here the sugar, and then we have the phosphate and the sugar and the phosphate. And in the middle here, what we see, those blue balls, blue structures right here, those are the nitrogen atoms. And we can see now that within they're found, the nitrogen atoms are part of, as we know now, they're part of the bases. And they are found right in the middle of the double helix structure. So that's how they are kept together. Now, if we simplify this structure that we've seen before, and we look at it spinning, now it's now even easier to see the interaction between the sugar and the phosphate here, known as the sugar phosphate backbone. Where I want to attract your attention to is right here in the middle here. So you see this part here, if I can make it spin in space a tiny bit and then maybe show you a better representation, I hope this way you'll be able to see it, right? So we have one base here, we have as you can tell, we have a pyrimidine right here, the simplest of the cycles. And then on the other side here, we have a hint of a purine molecule here. So pyrimidine, purine. And in this case here, we have a pyrimidine here and a purine there. And those two are interacting with one another. They are interacting this way and this way. And the way they are interacting is via hydrogen bonding. And every single one of those nitrogenous bases in the middle are kept together. They keep the strands, the sugar phosphate backbone on both sides. They're kept together via hydrogen bonding in the middle. And that is a very important feature.